So then we'll move on to our last talk in this workshop. That's from Melanie. And then we'll have some time for a bit of discussion, and then we can always come back to any of the other points, and if you have any questions that you've saved up. So Melanie is now at OICR in Canada, leading a team there. She was in the Seneca project with us. That's how we all know her. And of course, you've heard her name mentioned several times, associated with GECKO, IHCC, etc. So thanks, Melanie. We look forward to hearing about the GA4GH work.
Wonderful, thank you. It's really great to be here and see a lot of familiar faces and wonderful live demos. I'm afraid I do not have a live demo, so it may be a little bit less exciting, but this is also a little bit like Leslie's effort. This is a new effort from GA4GH. And I think today, I'd really like to, I guess, get your interest in possibly joining this group and helping us, define those standards that we do need for harmonization. So today I want to talk to you about work we are doing with the Computable cohort representation group in GA4GH. So for those of you who may not be highly familiar with GA4GH, and I don't want to go too much into details, but the website link is down there, so you can go on the website and get all the information. It's a gigantic consortium of, I'm going to say, coalitions of the willing. So it's people who are, convinced that we should all work together to set standard and specification to enable genomic data sharing at scale, and that's, doing that responsibly, minding ethics and regulatory restrictions, but also setting those standards for harmonization and consistent representation. There's a lot of cloud work for data sharing online, big data sets and things like that. And it's truly a worldwide effort. So of course, H3 Africa is in there, but there's also a representative in North America, South America, Europe, and also in Asia. What GA4GH thinks is that, by 2030, over 300 million genomes will have been sequenced. And unless we're able to bring those large cohorts filled with individuals from those different populations together, we're going to miss on that opportunity to build that virtual cohort of millions of people, which we can then use to do cool research. So that's really the goal of GA4GH overall, and the rationale for the products and standards that are being developed. So this is a very high-level overview of those goals in a little bit more details and how they pertain to each of the GA4GH standards. So you can see, if you start from the data donor, we have Your DNA, Your Say, which is a survey about what you think happens to your genomic data. There's work around consent, sequencing, ethics I mentioned. It goes to the data access committee where requests for access are being approved, then the researcher gets access to the data. Then there's all the work around finding data set for Beacon, which many of you may be familiar with, for example. All the cloud work is up there with the WES, TERS, TESS, and DRS API. And at the end, the goal is really improving human health and medicine. And to achieve this, and that's really just a little overview of the structure, we worked with, at the top here in red, real world driver projects. So, real projects that have real data. For example, in my team, I lead the ICGC Argo project, which is a cancer genomics repository. And on the left-hand side here in purple and blue, we work with technical work stream. And at the bottom here, those foundational work stream, around data security and ethics that I mentioned. In particular today, the very last blue block here, the clinical and phenotypic data capture, this is the work stream I co-lead with David Hansen in Australia. And this is where the cohort representation subgroup lives at the moment. So what did we do? We know there's a challenge bringing cohort data together, and we wanted to get a little bit more, I guess, background information from people trying to do that or people interested in doing that. And we did a landscape review first of resources that we could think about that could help us achieve cohort representation and integration. So, you can see the beacon at the top here. Seneca is in row 6. So that's the Gecko resource and the model. There's the HDR phenotype library, N3C in North America for COVID. So, we try to come together and think, okay, what are the resources we know of? The second step then was looking at what type of resources were submitted. So we had 6 submissions to our use case collection form as well. When we looked at the landscape of the submission, there's a lot of data model and application knowledge based, a little bit less on cohort registry and cohort summary and identifier systems. And part of the work that we want to do is really to highlight and address the pain points and requirements here and really target those practically. So, lack of interoperability. You just heard from Mamana show how we need to be able to harmonize attributes, things like demographic variables not standardized. It's hard to perform analysis when your baseline is different. The individual values are often not available or even shareable. And the requirement to address those is that we need interoperable cohort definition, and those should be computable. We want that computational description of the cohort. So this is one that we can run as the baseline for the population. And we want models that allow to represent those different sources and link consistently, to ontologies, data elements, vocabularies, and also those mappings should be available, ideally with additional provenance information, but at least the mappings should be available and reusable. So we decided, we came together as a group, we took that feedback really to heart, and we thought, okay, where are we today? So today we have examples from cohort discovery. So you've heard about, for example, the IHCC registry, where somebody could be looking, so that's on the left-hand side here, somebody could be looking at cohort with blood measurement data. And that relies on, standardization against the model, ontologies, defining things across cohort. It's using Gecko, it's very ontology driven, and that returns cohort. And we could link that to phenopacket, which is a standard structure for phenotype representation. On the right-hand side, we have other cases, for example, HDR UK, where they're looking more into the details of the measurements in the cohort. And that really needs standardization at the phenotype level. So we need to be able to know we're all talking about the same thing when we talk about creatinine level, for example. And that also means we need to be able to query at the individual row level and return numbers of patients of information about individual rather than cohort. So I think we're in the middle. We're the purple people. So we're trying to build that bridge between the two use cases, that cohort representation, which would contain metadata from the registry and the data set and the phenotype. So we would be able to link between both sides. As I said, we need to have those mappings also concretized and available. We want to reuse what's already been done, so link to N3C or OMOP, FKB for computational representation that are already out there, for example, for phenotypes. And we want to be able to do discovery and query-driven cohort definition and have those standards that, again, will link and cover both areas. So more practically, if you want a simple example, on the left-hand side, this is the blood measurement. You go to the HCC browser, you're looking for cohort that capture a specific type of measurement, like blood measurement, say, hey, found it, and it returns cohort A. On the right-hand side, so this is the example from, for example, HDR UK, where you have that one cohort with participants, and you're looking for female participants that have a specific lab result. So here, for example, creatinine above 0.9, and you're able to find a subset of participants in that specific cohort that would match against that query. So the cohort registry discovery, this is just an example where we have a researcher looking at the type of information that can be collected. So for example, In this case, we've collected information about 3 cohorts. Using a model like Gecko, we can know that blood, fasting, and creatinine are actually type of blood measurements. So a query looking for blood measurement data would return cohort 1 and cohort 3. So that would be the left-hand side use case in the table. And again, here the example of the HCC Atlas and how you can do those types of query with the faceted menu. The other hand side of the example is, again, we have 3 cohorts. We are looking for asthmatic patients with a creatinine above a certain level. So in this case, we need to be able to look at the type of lab results, and we need to be able to look at the diagnosis. And you can see that the diagnosis, right here, you have ICD-10 code and SNOMED city. You can see that the diagnoses do need to be harmonized if you want to be able to query against all of the rows. And that's the example of what HDR UK is trying to achieve. So the Atlas for cohort registry discovery, and HDR UK is looking at the computable cohort discovery. So within one cohort, using, for example, age here in the orange bubble, or measurement of some body and things like that. So you can, using that, you can search within one cohort for specific measurements. So this is work that's done by Sushil Varma, if you can see the name here in the top right-hand side. We want to be able to build a model that allows us, and I'm not talking about an ontology, I'm talking about a data model, which allows us to do that representation consistently. And to do that, we want to build first a minimum information about computable cohorts. If we want to build computable cohort, what is the type of information we need to capture? So, in the middle here, you have the cohort. Of course, you want to capture the phenotype, possibly the algorithm associated on the right-hand side, but you also want to capture, the study the cohort come from, who's running the cohort, and also cohort summary, if you can get, for example, to summary statistics for your cohort. So we're calling that MIACC, the minimum information about computable cohorts. And again, we don't want to redo things. We tend to be lazy, and we like reusing things that other smart people have done. So reusing standards like Gecko, you may be familiar with FIRE or OMOP, Phenopackets and Beacon, which are other GF or GH products. And so we think this is a proposal of what we think it could like. This doesn't exist yet. This is something we're striving to go towards. And moving forward, how we are going to work. So we had a long discussion. How are we going to make this happen? At the end of the day, the truth is that you need somebody to do hands-on work. So we thought, okay, let's look at what successful organizations are doing. And for example, Amazon is having those 2 pizza teams, which are teams of 5-7 people that you can feed with 2 pizzas. And those teams meet on a regular basis in some sort of mini hackathon. And they're formed with variety of users. So we have, people who have use cases, researchers, people who can do the technical implementation, people who have a platform where we can run those use cases. And so we've constituted 2 of those teams. So we constituted one team, which is going to deal with the right-hand side use case, which is working on, capturing the deep phenotypic characteristics as a standard. So we can use that consistently. And another team that's looking more around the data model, which is that minimum information about computable cohort. Initially, we were planning on having those running concurrently. That was just a little bit too much. So now we've moved over to sequential. So Pheno is going on right now, and MIACC will follow afterwards. So where are we? So we've set up those computable cohorts. So we are focusing on the Pheno aspect now. So now we want to show that exemplar phenotype across those different definitions. We've used asthma as a driving use case, and we want to try and see, can we actually do those federated query and test that across implementation. We split the hackathon into 5 different sessions. May 6th, we talked about, data access. We tried to get access to data. There were lots of problems. We thought this was an important lesson learned. Last week, we were talking about Beacon to access cohort representation. Today, actually, earlier today, there was a discussion around phenopackets and FHIR. And then next week and the following week, we'll talk about phenotype matching tools and FHIR and terminology servers. Where we're at, so the data has reviewed those data access for a process for these different databases. If you remember very early on the work stream, there is a work stream I'm also part of that's looking at data use and data access. So we'll work with them to isolate those pain points and try to address those challenges. The Cohort Beacon, so Jordi Rambla, who's also part of Seneca, presented the Beacon V2 servers and how that could be implemented. And I think that's really interesting, having a Seneca implementathon, which would be a practical Beacon implementer meeting. Come with your data and we'll help you set up a Beacon. There's different resources linked here, and the slides are available online as well. Future direction, and again, this is a call, please join us. Please reach out to Lindsay if you're interested in getting involved, if you have a different use cases, if you can see something we missed, we forgot. Send us an email, reach out to Lindsay, attend the meeting. It's really an open group. At the end of those 5 weeks of Hackathon, we'll have a global report back meeting. So this is going to be done by the clinical phenotypic workstream. And we need to think about way forwards. Do we think there is matter for that to become another product for GA4GH, and we should do a product proposal? Do we think if there's interest in having regular meetings and bringing that product forward, then we need to set those up. And as we move forward with Hackathon, I think we discovered that we're having much more issues than we anticipated. So there's going to be an interesting aspect of the work. Or can we resolve those action items? It's always the same. As soon as you try and do things in real life, there's things you didn't think of that pop up. This is a large group, so we've been really lucky that a lot of people have seen value in this work and have joined the group. This is a group I co-lead with Sushil Verma, but I should mention that Ian Forey has done a lot of the drive behind the implementation with Jordi, and Judith was looking also into the MIACC standards. So yeah, thanks to everybody here. And again, if you want to join us, please do. There's actually a GA4GH onboarding program. So not only for this work, but if you're interested in GA4GH as a whole, you can reach out, for example, to Nirja, and she'll put you in touch with workstream guides so that you have an easier time coming into the network. We're really interested in having new people come on with new ideas and participate. Thank you.
Thanks, Melanie. So that was also a fantastic talk. Are there questions for Melanie? This is a reasonably new initiative, but they've already gone quite far. So I think, as Melanie says, if you want to get involved, I think it's really useful to get involved now. Any questions?
If you have no questions, no, I'm happy. Did you have one, Nikki?
Sorry, yeah, the beacon. I'm not sure that everybody knows what a beacon is. So, so basically, you're taking the beacon implementation and seeing if you can use this for discovering those data from those minimal sets that you're going to be defining.
Yeah. So the current beacon, the V1, was only looking at genomic queries. So, do you have an allele with a specific variant? The Beacon V2, which I think has just been approved by GA4GH, actually includes the ability to do phenotypic like queries. So, do you have a female patient, for example? So, because we don't want to redo what other people have done, it does seem like there's an overlap there. So Beacon uses the phenopacket as a data model, as a structure. And we were thinking about using phenopackets to represent that cohort structure as well. Phenopacket as a core message, and the phenopacket group was really keen on, you know, adding to that message, which I think pretty much know is just a cohort ID and a set of individuals. So we're keen on expanding that as well. So it does seem like there's a marriage waiting to happen there. We're trying things, you know, it seems to work okay for now.
Yeah,
So.
I mean, I think it's sort of a difficult because so the coming in from the here's a particular position, you know, there's a very clear reference. So that coordinate is not going to be ambiguous. But if you're coming in from something that might be ambiguous, it might be harder to find what you're looking for, if you know what I mean, and know that everybody's representing it the same way. I mean, obviously that's what the standard is going to try and do. So I can see there's going to be a use for it, but I think it's going to be a bit more challenging than the original use of Beacon, which is coming in from a very defined set of coordinates that the whole world uses and, you know, everybody knows. So, but I think
no, that's right. That's so that's part of the discussion we had last week, and Jordi was showing those different ways to implement Beacon, and you know, because Beacon is if you want a super querier, so it comes in and say, give me everything that's female. Of course, on the other hand side, your resource needs to be able to say, what does female mean and what's female in my cohort, right? So somewhere there has to be a mapping. I don't want to go into too much detail, but the implementation that the Beacon group was recommending is a lot of resources already have an API. So, you know, you're probably able to come to a H3 Africa dataset and say, which are the female participants, and then your API understand what that means. So then having the Beacon API come on top rather than trying to convert your own API to Beacon, because that's impacting your own resource, which you may not want to do. So it's really about layering those APIs on top so that we can do those federated queries across several resources while not impacting the individual resources too much.
okay. Yeah, that makes sense. Any comments or questions from anybody else? Chats, everybody's saying very interesting presentation. Great overview of phenotype harmonization activities. You reminded me to sign up for the cohort representation group. Yes, I signed up for the MIACC one, and I was wondering why it was so quiet. So obviously because you're doing the other one first.
I know, as I said, we are really planning on having both groups running concurrently, and that was just too much. It's
complicated. Yeah.
A lot of people are interested in both, and it was just getting the manpower and the willingness on both at the same time.
Yeah.
Too much of a
sure.
commitment.
But you know, I found that when I was on the initial meetings, that it also helps for people who, you know, like on the workshops that are trying to do their own harmonization, because the discussions bring up some points, and I think each cohort actually brings in their own perspective. They bring in novel barriers, they're bringing novel challenges and stuff, and so it all contributes to really getting a more global representation of, you know, what's going to be developed. So I think it is important that there is sort of diversity in the group that comes in and contributes to that, even if it's sort of just adding briefly to the discussions. Okay, I don't see any actual questions apart from how interesting everything was.